I'm Dr. Sherman Silber from St. Louis, and I'm very excited to share this session of what we call the CHOSE project. And that's a very special collaboration between Dr. Hayashi from Japan and Dr. Clark from UCLA Stem Cell Center and Dr. Orwig from the University of Pittsburgh. I think these are the leading three people in the world on stem cells, converting stem cells to germ cells. And we do think that is the future based on Dr. Hayashi's original studies in the mouse. We have to believe this is going to be a game changer in the future of IVF. I'm just a clinician. They're the scientists making this possible, but I'll just make a brief introduction and introduce the speakers as we go. We'll each talk for about 15 minutes. And my major clinical point is I've learned from the in vitro gametogenesis work of Dr. Hayashi, as well as Dr. Clark and Dr. Orwig, how to do IVM. It's very robust, and I'm not the only one doing it, but we really understand why it works now, and it's really spectacular. So I'll go into that. Now, so this introduction to the CHOSE project will focus just for 15 minutes on IVM. And this is the CHOSE team originally, and you can see us all there at the UCLA Stem Cell Center. This is my first work with Katsuhiko Hayashi in Japan at the University of Kyushu. And this is Dr. Hayashi with Dr. Hikabe. Ori Hikabe was the first author on the incredible paper in which they went from skin cells in the mouse all the way to healthy baby pups. This is Dr. Clark, who is really, I'm going to embarrass her, but she's really the world's expert, I think, on applying this to the human. And that's the whole object of the CHOSE project, is to apply what Hayashi could do in the mouse with his advice to the human. And this is Kyle Orwig, who is well known, quite specialized for the one person Dr. Hayashi said in the world that we should collaborate with is Kyle Orwig for working on making sperm from skin biopsies. And these are technicians that helped us with this work, Sierra Goldsmith, and I don't have Tim's name down here, but it's a much larger team than this, but these were the originators. So I'd like to just make a simple point about the CHOSE project, that in the mouse, and this, some people don't know all this, so I'll try to simplify it. In the mouse, we first make iPS cells, which are like embryonic stem cells, from skin cells of the mouse's tail. Those iPS cells are then transformed to PGCs. Now, you incubate these primordial germ cells with fetal granulosa cells, not adult granulosa cells, only fetal granulosa cells, to get fully competent oocytes in normal baby mice. Applying this to the human now, with Amander, I say we, but it's Amander, now has many beautiful human PGCs derived from a simple skin biopsy from our menopausal women. And now we're at the stage where we need to make human fetal granulosa cells, which Dr. Hayashi is helping us with, so that we can then wind up making competent oocytes from these PGCs. This is the biopsy. This is about all we do. It's pretty simple surgery. We just do a simple punch biopsy and we send it to UCLA. And being considered for publication is our first series of identical twins. And we wanted to find out what stage that a PGC or germ cell specification occurs. It was kind of an offshoot of this. And you can see we're successful in making PGCs from menopausal women. And in fact, we have one 55-year-old woman who was previously fertile who generated what we call young PGCs from 55-year-old skin cells. And so just think about this. Once we perfect this in the future, a 38-year-old woman with 38-year-old eggs might be better off having a skin biopsy than an oocyte retrieval for IVF. So I'll talk about in vitro maturation and how we've been able to really astoundingly make this robust just using Hayashi principles. And the most important thing is not to do the wrong thing, because it's so simple. Any ordinary IVF culture medium is fine. IVM media is absolutely no better than any non-IVM media. It doesn't matter what your media is. Human serum albumin, of course, And then we would use 150 milli-IU per ml of HCG, but any other concentration from 10 milli-IU to 1,000 milli-IU works equally well. The same is true with FSH. 150 milli-IU per ml, that's what everybody talks about, sounds physiological, but frankly, any other concentration is just fine. And frankly, you can just take Menopur, which I'm not advertising, I don't even like the drug, but you can dilute it 1 to 1,000, and that gives you 10 milli-IU of HCG and 75 milli-IU of FSH per ml, and that works fine too. So it's amazing how automatic this and simple it is. So this is how we do it. This is only going to apply to ovary tissue. When we take out the ovary, in order to freeze ovary tissue for fertility preservation, we take out the whole ovary. And work by Yasui et al. From Japan has shown there's no question that taking out one ovary does not make the donor any less fertile or likely to have her menopause more than a year earlier than the average age of 51. So do not fool around with just taking out a little bit of the ovary, because then you ruin your chance to get these GVs. And we just dissect it the way Roger Gosden originally indicated. Here you're seeing we're dissecting it on ice. We get a little better results if we dissect it really at room temperature. And there we have the nicely dissected ovarian cortex that's thin enough that we can use it for freezing and transplantation. And we cut it up into pieces because, you know, when we do the freezing, it's not just the thinness of the tissue, but the volume of the tissue that affects the rate of cooling. And so we know from all the other studies that it can be 1 or 2 millimeters thick, but it has to be no more than 1 by 1 centimeter square. And there they all are ready to be frozen. But then we don't throw it away. We look at the spent media. And it's amazing when you look at the spent media. It's just incredible how many cumulus complexes we see. Now, this is just histology to sort of show you what's going on. When we dissect across that line of demarcation of the cortex, at that very line, there are many, many follicles that are still microscopic. You really don't see them, but they contain GV oocytes. And there's what that looks like the first day. And you don't want to remove the cumulus. We've done studies where we remove the cumulus right away, and that's a disaster. It still needs the cumulus. But there you see 1 day post incubation in any combination of HCG in any media. Really, you see beautiful metaphase 2 oocytes. And here you see actually 3 of them with 2 that did not mature. And here you see 3 that did mature with 2 that degenerated. But that is just a recapitulation of what normally happens physiologically. And that's the secret to what we're doing. So you can see with a big slide like this, no matter what FSH or HCG concentration you use, no matter what media you use, IVM or cleavage media, it doesn't matter. We average about a 35% maturation rate to normal M2s. Unless the cumulus is stripped immediately, then it's a disaster. So many mature oocytes can be obtained from ovary tissue with simple media and no need for ovarian stimulation, but the intact cumulus is crucial. And this isn't just us. Klaus Anderson has the same result. Seegers has the same result. And even in the mouse, Hayashi has the same result. Now, Anderson did go further and did NGS sequencing of chromosomes on the M2 oocytes retrieved this way, and 64% were euploid, which is about a normal incidence. So the M2 rate is 1 in 3, and was resulting in the same number of M2 oocytes that you would get just with ovarian stimulation. And we know that it really works. We haven't had a chance to put these embryos back yet in people, but Seegers has already shown that these are viable embryos that result in healthy babies. So why does it not require special IVM media, and why does IVM work? Well, in a way, it's easy. You don't have to really know anything else, though I'll explain a little bit more in a minute. GV oocytes dissected from ovarian tissue have already become meiotically competent in vivo by what we term, and this was Hayashi's original terms, IVD and IVG, in vitro differentiation and in vitro gonadotropin sensitivity. So they have to become meiotically competent with IVD, but then they have to develop their competence for maturing by the usual 11 days exposure to FSH, the same in vitro as is in vivo. But also it's far easier to obtain many GV oocytes from tiny follicles. You can't see them, opening by open cortical dissection rather than by using a needle. So to understand why IVM works and works so easily, you just have to understand in vitro gametogenesis. So this is my favorite slide. You know, the Japanese are supposed to be short and Americans are supposed to be tall. So Hayashi is the X chromosome and I'm the Y chromosome. But it all begins here at the epiblast with PGC specification. And actually, you know that all the resting follicles are located in the outer cortex, and then they gradually mature and grow towards the soft inside. So Hayashi's shown you need low pressure and you need 8 core genes for this recruitment from primordial follicles to occur. And this little diagram, you can see the IVD phase is where the oocyte first enters meiosis. You have to have that, and you need granulosa cells to do that. But then once they become secondary follicles, then that is the moment that they are sensitive to gonadotropin, and that's the IVG phase, which is usually 11 days. And after that, just 1 day of exposure to HCG results in conversion from GV to metaphase 2. So this is a long story about how Hayashi originally was able to just make competent eggs in baby mice from skin cells. But if you look at the last 3 lines, you'll see the PGCs are incubated with granulosa cells for 3 weeks in the mouse, and that's IVD. There is no hormonal sensitivity at this stage, and it's equivalent to the first 4 months after recruitment of primordial follicles from the human. And then you just need FSH, and it's amazing, the same as in humans, an average of 11 days. And then you can culture with HCG for IVM. Now, one thing I've got to tell Dr. Hayashi, I think that the FSH culture should add a small amount of LH also, because we know that's important in human ovarian stimulation, and we might get more efficiency that way. Here's Dr. Hayashi and Hikabe's original diagram, and you can see how important it is to understand IVD, which is not hormonally sensitive at all, and then IVG, the moment that those secondary follicles become sensitive gonadotropin. So during the first 3 weeks of culture of PGCs and fetal granulosa cells, this is IVD, so there's no FSH. Then you open and culture those follicles open in FSH, that's IVG. And as I said, maybe we should add a little LH to the FSH during IVG. So this is what it looks like when he's made this, so to speak, artificial ovary with fetal granulosa cells. And then these are the first iPS cell mouse pups, healthy pups that were born just from the skin of the mother. So why does IVM work? Well, many GVs are inside the medulla. They've had adequate time of exposure in vivo to FSH. That is because there are so many of them, 30 per day, that were recruited from primordial follicles 4 months earlier. So we get 30 per day that are entering that phase. It is much easier to collect many GVs from these tiny follicles with the ovary in hand instead of using a needle. And 35% of these Gs have already been made meiotically competent from in vivo exposure to FSH for 10 to 14 days. So oocytes in the human ovary 4 to 5 months after recruitment have completed IVD and IVG and are ready for IVM. It's a constantly changing daily dynamic. In vivo, once the oocytes have completed IVG, they are meiotically competent and ready for HCG, and this is a constant daily process. So my time is running low, so I'll just a couple more explanations. Secondary follicles that just finished IVD are now gonadotropin sensitive. And so these are the follicles we actually enlarge with ovarian stimulation, but you don't have to enlarge them. They're exposed to FSH anyway in the ovary, in vivo. New secondary follicles are appearing every day, an average of 30 per day after finishing IVD. GVs that have finished the IVG are now meiotically competent and ready for HCG. So in an ovulatory cycle where one lead follicle becomes dominant, the non-dominant follicles still are going through IVG and attain meiotic competence. So the question, this is even why many IVF works so well, because we just want the GVs that have just become gonadotropin sensitive. Those that have not become, those that have not been exposed long enough to IVG won't be very good. Those that have been exposed to too long won't be good. So we put them on birth control pills to suppress all those, and we start only with the ones that have just entered the sensitive phase. So the grand conclusion of this is you do not need ovarian stimulation for oocyte maturity. FSH has already created GVs with meiotic competence, and it's happened in vivo for us. Do not even need the normal ovulatory cycle for meiotic competence of GVs. We only need ovarian stimulation or the ovulatory hormonal cycle for follicle enlargement and either normal monoovulation or for oocyte retrieval. So in vitro gametogenesis and IVM reveal the limited role of the ovulatory cycle and ovarian stimulation in oocyte development, other than simply to exit the oocyte from the ovary. And this is when they were, when I went to Kyushu on my birthday, they celebrated my birthday. It was the greatest birthday party I can ever remember. I believe that was my 70th. But now I'd like to have Dr. Hayashi present, and he's going to give us much more detail about his groundbreaking work in the mouse, which started all this going.
Great. So thank you for Sherman for very kind introduction to our study. So today I'm going to introduce actually how contribute of this mouse project to the CHOSE project that is going to make a sperm and egg by the Kyle and Amander. So I am trying to establish kind of the proof of concept in this gametogenesis by using a mouse. So I'm going to introduce very briefly about how we can make especially egg from pluripotent stem cell in the mouse. So we have been working on the in vitro gametogenesis where we can get oocyte and sperm from pluripotent stem cells for almost 10 years. Then this kind of in vitro gametogenesis would be efficient tool for the better understanding of gametogenesis, which is very complicated process. And then also it's going to be, as we aimed, alternative source of the gametes for the research and reproduction. So let me very briefly introduce how germ cell is produced in the embryogenesis and in vivo. So all of the sperm and egg are derived from the PGC, pyramidal germ cells, that are specified at very early embryogenesis. Then, although they are indifferent between male and female, but they do make a difference in the gametogenesis depending on the environment of the gonads. So, of course, as everyone knows, when they are reached to the testicular tissues, then they undergo spermatogenesis, and then when they go into the ovarian environment, then they go oogenesis. So almost 10 years ago, we established in the mouse, make the primordial germ cells from pluripotent stem cells, including iPS cells. So as long as they culture under the undifferentiated condition, they keep self-renewing. But once they change into some culture condition, they acquire the competence to give rise to the primordial germ cell-like cells. Then upon the BMP4 stimulation, that is the inducer of the primordial germ cells, then they become the PTC like this. So the fluorescent cells are the primordial germ cell-like cells. Those are competent because when they're transplanted into testis, they give rise to functional sperm, and then they transplant into the ovary with the ovarian fetal ovarian somatic cell, then they become functional oocytes. That's almost 10 years ago, work in 10 years ago. So then with the Dr. Hikabe, as the Sherman introduced, we establish culture system to make the oocyte in culture without need of the transplantation. So in this system, the trick is to make the ovary organoid, where the ovarian environment can be kind of reproduced in vitro culture. So those primordial germ cell-like cells from pluripotent stem cells are deaggregated with ovarian somatic cell, right? Ovarian somatic cells. Then we make a kind of ovarian organoid. So, and then as Sherman introduced, this culture system can be divided into the three periods. In the one is IVD, where the PGC give rise to secondary follicles. Then IVG, where they give rise to the mature oocytes that they acquire the meiotic competence. Then in the IVM, they resume the meiosis, and they become the fertile egg. Then in the IVD culture, after 3 weeks 3 weeks later, they give rise to secondary follicles, and then IVG, they grow up to the mature oocyte, then acquire the meiotic competence. They indeed resume the meiosis and get fertilized with the wild type sperm. Then those fertilized eggs give rise to the two-cell embryos, then give rise to the pups after transplantation into shoot programmed females. So of course, the percentage of this producing the pups is not so high as the in vivo, but as Sherman said, maybe we can refine this kind of culture condition by controlling the hormones or some culture conditions. But at the moment, we can get the 3.5%. Maybe currently we can get 5% of the transplant embryos can give rise to pups. Then since then, people trying to apply this kind of system to the human, or not only the human, but also the other wild animals. So we are still in the middle way of this system, the technology to the apply to the kind of clinical level. So actually there is a big obstacles to apply this system to the other animals. That is actually gonadal somatic cells, because we use the embryonic gonadal somatic cells. That is the only materials for the maturation of primordial germ cells into the oocyte. So as long as we use such embryonic tissues, that is really difficult to apply this system, because it's really difficult to get such materials from for example, human embryos. So now we establish the culture system to introduce, to make the ovarian gonadal somatic cells, fetal ovarian gonadal somatic cells from pluripotent stem cells. So we don't need actually embryonic tissues. So let me introduce this system. So okay, so to make such the ovarian tissues, we should pay attention to the embryology, how the gonadal somatic cell is produced in embryogenesis. So this is the introduction of the how the ovarian somatic cells is produced in embryogenesis. So those tissues are derived from mesoderm, and then differentiate into the intermediate mesoderm. Then they give rise to the ceramic epithelium, that is the precursor of the gonadogenital ridge. Then they become the ovary. So to make the such a differentiate, to reconstitute this differential system in culture, we gave several kind of the factors that induce the differentiation in each step. So let me introduce each stage of the differentiation process. So we first try to make the nascent mesoderm by giving the BMP4 and Wnt signaling. So actually, to make the nascent mesoderm, the BMP4 and Wnt signaling have a synergistic role to differentiate such a cell type like this. Then in the second step, we need actually differentiation of the intermediate mesoderm. Actually, to make the intermediate mesoderm, the BMP4 concentration is really important, because the only at the intermediate concentration of the BMP4, we can get efficient differentiation of the intermediate mesoderm. If you give the excess level of the BMP4, then you get the lateral mesoderm that is more lateral side of the body. So the BMP4 is really important. So after making the intermediate mesoderm, what we need is we need to specify the genital rich ceramic epithelium at the genital rich region. So to specify such a cell type, we need to give some signal that control the body axis, the body plan. So by giving several factors that control such a body axis, we culture the several concentration, several combination of these factors. So actually, in some culture condition, where in this case there is the FGF signaling inhibitors and retinoic acid and sonic hedgehog, we can get such a genital rich region that expressed GATA4. So this is in vivo. You see the expression of GATA4 at the precursor of the ovary, and then you see also the GATA4 expression in the in vitro or aggregates that is derived from pluripotent stem cells. Then we keep culturing. Then we see almost spontaneously the ovarian precursor cells that express NRR5A1. So we call this fetal ovarian somatic cell-like cells for cell C, then see whether those are really functional. So then we check the gene expression profile by single cell sequencing analysis. And then this is the host LC derived from pluripotent stem cells. And those expression pattern of the fetal ovarian gonadal somatic cells from in vivo. So the pattern is almost same, although the proportion of each cell types are a little bit different. So actually, the differentiation of granulose cell type are a little bit inefficient compared to the in vivo. However, if you compare the gene expression profile of each cell type between in vitro and in vivo, those are almost identical. So we are almost confident that those are the function. So we de-aggregate such a post LC with the PGCLC, both of which are derived from pluripotent stem cells. So as you can see here, the ovary follicles are formed that express the germ cell markers and granular cell markers. And then they indeed undergo the corrective sequence of the differentiation process from PGC to the secondary follicles, including the entering meiosis like here, and follicle genesis. Then if you culture such artificial follicles, you see they grow up to the mature oocytes, mature follicles, with the proliferation of granular cells and then growing the oocyte. And you see also the transzonal projection between the oocyte and the granular cells. Then in the IVM, you get the mature oocytes, then they undergo the meiosis. Then when we use the IVF, then some of the oocytes get fertilized and become two-cell stage embryos. Then when they are transplanted into the pseudo-pregnant females, they indeed gave rise to pups. Then those pups give rise to the adult the mouse, and then those are fertile. So now we have like 5% of the two-cell stage embryos derived from artificially made follicles give rise to pups. So let me summarize my talk. So at least in the mouse, so we can get both germ cell lineage and also ovarian somatic cell lineage. Then by aggregating with both cell type, we can get the functional follicles and we can get the functional oocytes. So now, because we don't need actually embryonic tissues, now is the time to apply this system to the other animals. So, and also I'd like to stress that this kind of the culture system would be useful to investigate the oocyte and somatic interactions, which is also very complicated, as well as the gonadal somatic cell development. That is the very important for sex determination in the individuals. So thank you for the listening, and thank you for this collaboration.
That was awesome. I am so excited, and I'm anxious to hear what Amander Clark has to add, because I mean, this is the #1 issue. It's our holy grail, is to try to make oocytes or sperm from in humans or in endangered species that don't have it. And the big issue was fetal somatic ovarian cells, at least for eggs. And so now we've got the roadmap from you about how to try to do that, because otherwise you're not going to be able to find fetal granulosa, ovarian granule, or fetal ovarian somatic cells. Well, great. So now I'm going to introduce Amander Clark, who's she's really amazing. She was originally introduced to me by John McCary, and as this idea of the CHOSE project came together, it was obvious that she would be the key to applying this to the humans. She always amazes me, coming from a tiny little town in the middle of nowhere in Australia and wanting to be a veterinarian, becoming a giant of science. So I hope I didn't embarrass you too much, Amander, but take it away.
Thank you so much, Sherman. Actually, probably very few people knew that's what I was planning on doing after growing up on a farm. I wanted to be a veterinarian, and instead I fell in love with science. So thank you so much for the introduction. I'm going to talk today about generation of germ cells in humans from skin biopsies as part of the CHOSE project. So today, what we've been talking about is the formation of gametes, and Katsuhiko has very nicely introduced the development of germ cells in the mouse, starting from the early post-implantation stages of mouse development. What we now appreciate in humans is that germ cells form from a different place than they do in the mouse, and they start to be specified in the early post-implantation human embryo as the epiblast is distinguishing itself from the amnion. And so in humans, germ cells are first observed within the amnion, and then they migrate into the embryo and ultimately into the testis and the ovary that form a little later in embryo development. So what we're very interested in is developing an approach for inducing germ cells in vitro instead of studying germ cell formation in vivo in the embryo and ultimately the formation of gametes. So that leads us to the technology of in vitro gametogenesis. So the original version of this technology is starting with pluripotent stem cells that originate from blastocysts. So blastocysts are consented to research from the IVF clinic once an individual or couple has finished their family. And then scientists can use these leftover blastocysts to derive embryonic stem cell lines. These embryonic stem cell lines serve as precursors to differentiating every cell type in the body, including cells derived from ectoderm, endoderm, mesoderm. They can even form some extra embryonic cells, such as the amnion, for example. But we also appreciate that embryonic stem cells can be used to form germ cells. So this technique of in vitro gametogenesis using embryonic stem cells has been very important for us to understand cell and molecular principles of early human embryonic lineage formation, including the formation of germ cells. In vitro gametogenesis from skin biopsies adds a whole new dimension to being able to use in vitro gametogenesis for various purposes. So this is possible because embryonic stem cells derived from the blastocyst, which are pluripotent and are able to form every cell type in the body, are equivalent to a cell type that scientists make in the lab called induced pluripotent stem cells. And the induced pluripotent stem cells are also able to differentiate into every cell type in the body. Induced pluripotent stem cells are made from somatic cells. And in our case, what this involves is the consent of a biopsy, which Dr. Silber showed you in his talk, that my lab derives skin fibroblast cells from. And then using these skin fibroblast cells, we reprogram them with Sendai viruses that carry transcription factors, including KLF4, OCT4, and SOX2, that will then infect the fibroblast cells and deliver these transcription factors to promote reprogramming to create induced pluripotent stem cells. Critically, Sendai viruses are non-integrating. So what these viruses are doing, they are delivering the cargo. The cargo are the transcription factors, and these transcription factors are then transcribed and translated by the cell, and therefore the cell induces its own reprogramming following infection. Then the Sendai viruses are diluted out of the cell. This makes the induced pluripotent stem cells that are now in the germline, and we use these induced pluripotent stem cells to differentiate germ cells. So currently, we use in vitro gametogenesis from human-induced pluripotent stem cells to understand germ cell development, to understand reproductive disease, the disease of infertility, which I'm going to give you a few examples of today. And we also use this to study epigenetics and gene regulation. And that's because germ cells undergo predictable and very reliable epigenetic reprogramming. And so we can use germ cells to understand the molecular events involved in reprogramming. Future uses of IVG will include using IVG-derived germ cells to fertilize, to make embryos, and we can use these embryos for research purposes to understand embryo formation. We could use IVG-derived embryos to understand embryo-uterus interactions. And in the future, once this technology is proven safe, the in vitro gametogenesis could be used for reproductive purposes to help the infertile. The first step in vitro gametogenesis from skin biopsies is appropriate informed consent. And that's something that we've worked very hard on at UCLA and together with Dr. Sherman Silber and his technical staff. Informed consent requires a few specific comments in order to perform IVG. The first is that informed consent must stipulate the intent to make stem cells. So this is a recommendation by the International Society for Stem Cell Research that creates guidelines for stem cell research around the world. And so for a scientist who wants to make iPS cells from a patient's sample, whether it be a skin biopsy or another type of cell, for example, blood cells, the informed consent for this must include a comment that stem cells will be made from the biopsy. If the intent is to make embryos from IVG-derived germ cells, that also must be stipulated in the informed consent as well. For scientists that are making embryos following IVG, this requires a specialized scientific and ethical oversight review. This review is more than a typical IRB review. And in the US, where I'm located, this involves our institutional stem cell research oversight committees. In other places around the world, it could involve a national body, such as in the UK, it involves the HFEA. And in other places, it involves the IRB bolstered with specific expertise about IVG and embryo formation. So after going through this process in the US, you saw an image of a participant from Sherman Silber's talk who had donated a skin punch biopsy for reprogramming to make germ cells. So this is what that biopsy looks like when it arrives at UCLA. What we do is to put that biopsy in culture and we grow fibroblasts from the biopsy. And this is an image down the phase contrast microscope of what the fibroblasts look like after they grow out from the biopsy. It takes anywhere from 3 to 5 weeks to be able to collect the fibroblasts that we then can cryopreserve under liquid nitrogen. We thaw out these fibroblasts and we authenticate them using single nucleotide polymorphism, and we use this same approach to karyotype the line. What this does is it creates a forensic fingerprint of all of the cell lines that we have in our lab that have been consented to this research program. We then perform exome or whole genome sequencing in order to look for infertility genes that could be the source of infertility in the patients who are consenting to this research. And then finally, we make banks of human IPS sublines from every patient. And the goal is, in this research, is to make 3 fully characterized IPS sublines from every patient that consents to this research project. So far, Dr. Silber's clinic has consented almost 40 different research participants to our infertility study. Specifically, he's consented around 10 patients who have non-obstructive azoospermia and around 30 patients who are consenting to the ovarian insufficiency research projects. And this highlights that Sherman is really paying attention to race and ethnicity of the research participants, because we all appreciate that infertility is not a disease that affects just people who are white, but instead it affects everybody equally. I want to focus now on our research participants that have non-obstructive azoospermia, because we've made the most progress on these samples. So I told you we have around 10 patients that have been consented to this research project with non-obstructive azoospermia. The majority of them have been diagnosed with Sertoli cell-only syndrome. We have a patient that's been diagnosed with oligospermia and 2 patients that have been diagnosed with maturation arrest. One of the first things that we did was to collaborate with Frank Tudelman at the University of Munster in order to sequence the exomes of our male infertility patients to see if we could identify any new male infertility genes. Fortunately, one of our patients that was sequenced was found to have a potential pathologic mutation in a gene called MSH5, which stands for mut S homolog 5. This gene had been associated with infertility in POI patients, but had not been associated with infertility in male patients. So this was a new discovery. In our patient, as part of our cohort, we had a mutation that occurred at nucleotide position 964 that led to the transition of a cytosine base to a thymine base in this patient's DNA. A CT transition in the genome is actually very common. It's one of the most common transitions that occur, a common mutation that occur in humans. The CT transition in the core domain leads to a single amino acid substitution from an arginine to a cysteine. Critically, very little is known about the core domain of the MSH5 gene. So this patient sample now creates a very unique opportunity to understand the consequence of an arginine to cysteine base substitution in the core gene of MSH5 that we are now currently pursuing. Our procedure for differentiating germ cells from the IPS cells that we make from our research participants is shown here. It begins with the patient's IPS cells. We then differentiate these into germline progenitor cells in the presence of activin A and a Wnt agonist for 24 hours. We then take these cells off the plate. This is a 2D culture, and then culture them in 3-dimensional aggregates where germ cells are induced within the aggregates. And we can see the germ cells here forming in clusters within a single aggregate. We do this in 96-well plates, so you can imagine that every well of a 96-well plate has one aggregate in it, and within these one aggregates, we have clusters of germ cells called primordial germ cells. We're able to FACS sort these germ cells from the aggregates using cell surface markers. We don't need to genetically modify our iPS cells to isolate the germ cells, and you can see a robust germ cell population here. By using single-cell RNA sequencing of whole aggregates, what we've been able to show is that the germ cells we make in vitro from iPS cells are equivalent to newly specified germ cells. I know this is hard to read. These are newly specified germ cells that have been identified in a human embryo in vivo at Carnegie stage 7, and that our germ cells we make in vitro are very similar to these cells and are distinct from the fetal stage germ cells that are found once they've arrived in the gonad and started to respond to the niche. So our lab and labs around the world are very good at making these early germ cells called primordial germ cell-like cells in the lab. The next step is, how do we promote germ cell maturation? So what you heard from Katsuhiko was that he has developed approaches for differentiating fetal gonadal somatic cells that he then combines with the germ cells that he makes in vitro from induced pluripotent stem cells to make a reproductive organoid. Within this reproductive organoid, we have the maturation of germ cells into gametes. So what our lab has been doing is learning this approach from Katsuhiko so that we can recapitulate it in our own lab. And we find that his approach for making gametes by combining together germ cells, the primordial germ cell-like cells, with fetal somatic cells of the ovary into these organoids, that we too can very consistently and reproducibly make gametes within the organoids. So in blue is the oocyte that's derived from the primordial germ cell-like cells, which carry a transgene that lights up blue under light. And then in gray are the somatic cells that in this particular case are the somatic cells that came from a mouse embryonic fetus. We know that organoids will promote the maturation of the primordial germ cell-like cells. So this is work that was published in 2018 by Mitnori Saito's lab, where he took primordial germ cell-like cells made from induced pluripotent stem cells and combined them with mouse embryonic somatic cells. So this makes it a xenogenic ovarian organoid, which he then cultured for up to 120 days. And within this organoid, after 120 days, he was able to see the maturation of germ cells by the turning on of this marker DDX4 in the iPS-derived germ cells, and the beginning of clustering of the FOXL2 positive granulosa cells around these oogonia that are differentiating in organoids. Because our research project involves not just forming oocytes, but also forming sperm, it's really important that I show you that testicular organoids can also be made. So xenogenic testicular organoids were recently published by Yang-san Huan in Nature Communications. And he also is able to show, using the same principles, that spermatogonia, fetal stage spermatogonia, but nonetheless spermatogonia can be produced within these organoids that start turning on the gene PWE-like4 after 124 days. In conclusion, in vitro gametogenesis from skin biopsies, we believe, is a personalized approach for understanding infertility. It requires informed consent to be able to perform these experiments in the lab. And we believe it's an important process for identifying and studying pathologic mutations from infertile patients. And we've already shown that we're able to identify new infertility genes using patient samples, and then take that one step further and use the patient's iPS cells to understand what that pathologic mutation is doing to affect germ cell development. And finally, organoid technology is necessary to mature the germ cells into either oogonia and spermatogonia towards the formation of gametes. And with that, I'd like to thank you all for listening. These are the people in my lab who perform the experiments. I'd like to thank the Luca Bella Foundation, who funds the CHOSE project, and my collaborators, in particular, Sherman Silber from the Fertility Center of St. Louis, Katsuhiko Hayashi, as well as Kyle Orwig. So thank you very much.
Thank you so much, Amander. As usual, that's really stunning. And I'll have questions later, but I have many of them right now. Next, Kyle Orwig is going to make the final presentation before questions and answers. And of course, he's very famous for working on fertility preservation in the male, actually. And I'm not sure if he's going to cover that. Amander sort of alluded it to with testicular organoids, but whatever he covers on stem cells and reproduction is going to be fascinating. So it's up to you now, Kyle. You're on the home stretch.
All right. Thank you, Sherman. And I'd like to thank the other organizers for the opportunity to speak here today. The title of my talk is Stem Cell and Tissue Transplant Therapies for Infertility. And of course, this is part of the CHOSE project. Well, I'd like to start out by introducing you to somebody that you all know very well. This is Louise Brown. She was born on July 25th of 1978 by the technology of in vitro fertilization. And she's shown here holding the glass jar, which is the incubator where her dad's sperm were introduced to her mom's eggs. Now, Louise was possible because her mom and her dad were able to produce sperm and eggs. The reason that they were infertile was because her mom had a blockage in her fallopian tubes. But unfortunately, there are a full 1% of men in the global population who are azoospermic, which means that they have no sperm in their ejaculate. And similarly, there are 1% of women in the global population who have the condition of premature ovarian insufficiency or no eggs. In the United States, those numbers translate to 1.3 million people between the ages of 20 and 50 years old. Now, the causes of these most substantial cases of infertility are several. They can include aneuploidies, such as Y chromosome deletions, or XO of Turner's, or XY of Klinefelter's. They can include smaller genetic mutations, including single nucleotide polymorphisms, like the one that Amander described, or small insertions or deletions. Or they can include medical treatments like chemotherapy or radiation treatments for cancer, or conditioning prior to bone marrow transplantation for non-malignant diseases such as sickle cell disease or beta thalassemia, or treatments for gender dysphoria. And for storytelling purposes today, I'm going to focus on the medical treatments that can cause infertility. So for adult men or teenage boys to preserve their future fertility, if they're at risk due to the medical treatment that they're going to get, they have the option to cryopreserve a semen sample with sperm, because we know that those samples can be thawed in the future and used to achieve a pregnancy with partners' eggs using the in vitro fertilization technology that was used to produce Louise Brown. But unfortunately, those options are not available to prepubertal boys who aren't yet producing sperm. And so for those patients, we have the option to cryopreserve pieces of immature testicular tissue. This is an experimental approach because we believe in the future it'll be possible to thaw those tissues out and mature them to produce sperm either through in vivo or in vitro approaches. And there are a variety of promising approaches in the research pipeline that can be broadly divided into cell-based therapies or tissue-based therapies. And today I'm going to focus on two approaches that I believe are mature and ready for the clinic. In fact, we're approved now to do both of these methods in our clinic in Pittsburgh. And those approaches are spermatogonial stem cell transplantation and autologous testicular tissue grafting. Before I leave this slide, I think it's appropriate to say in the context of our CHOSE project that one day it may not be necessary to cryopreserve cells or tissues before a gonadotoxic treatment, because one day it might be possible for a patient when they're ready to start a family to have a skin biopsy taken to produce induced pluripotent stem cells and then in vitro gametogenesis to produce either transplantable stem cells or sperm in a petri dish. But for today, I'm going to focus on the two transplant technologies that could be performed using cryopreserved prepubertal testicular tissues. So I'll start out by telling you about a model we developed in the laboratory several years ago. It's what we call a monkey model of cancer survivorship. And this was work that was done by Brian Herman when he was a postdoctoral fellow in the lab. In this model, we treated rhesus macaques with an alkylating chemotherapy that we knew would cause infertility. But before we did that, Brian took a biopsy from the testes of these animals and cryopreserved that immature tissue biopsy. The animals were then treated with chemotherapy, which caused them to become infertile. So after they survived the chemotherapy treatment, we thawed out their cryopreserved testicular tissues, digested them with enzymes to produce a suspension of cells, and then we treated them with a lentivirus that introduced the lentivirus and GFP genome into the host cells. In that way, we'll be able to tell if sperm ejaculated from the recipient monkey came from the cells that were frozen and thawed and transplanted. So here I'm showing you the results for one of those monkeys. You can see that he was making sperm prior to his chemotherapy treatment. After he received his chemotherapy, his sperm counts went down to 0 about 10 weeks later. So at that time, we thawed out his testicular tissues and produced a cell suspension that was transplanted back into the seminiferous tubules of his testes. And about 30 weeks later, you can see that sperm counts began to rise into the normal range for a peripubertal rhesus macaque. And we know that at least some of those sperm came from the frozen, thawed, and transplanted cells, because when we did PCR for the lentivirus and GFP gene, we could see a band. And of course, that band wasn't present in the pre-transplant ejaculates. I won't have time to show you the data today, but we did go on to show that these sperm are functionally competent to fertilize rhesus oocytes. We did that by sending sperm out to our friends at the Oregon National Primate Research Center in Oregon, where they have a monkey IVF laboratory. And they showed that these sperm could fertilize monkey oocytes by intracytoplasmic sperm injection. And this led to pre-implantation embryo development up to the blastocyst stage. We haven't yet produced a pregnancy or offspring using this approach, so that's the focus of our ongoing studies. So the other approach that I told you was mature and I believe ready for the clinic today is the technique of autologous testicular tissue grafting. Again, we return to our monkey model of cancer survivorship. This is work that was done by Adetunji Fayomi when he was in the lab as a PhD student, and Karen Peters, who was a research associate, and actually she's now a nurse on the cancer floor at our institution. Again, a biopsy was taken from the testes of these animals and cryopreserved. The animals were then treated with chemotherapy to make them infertile. And after they recovered the chemotherapy, we thawed out those tissues and transplanted them back into the animal. In this case, it's an intact piece of tissue. So the stem cells in the testis are still maintained within the seminiferous tubules of the tissue. And the objective here is not to restore normal spermatogenesis within the seminiferous tubules, but rather it's to graft the tissue under the skin and allow the in vivo environment to mature that tissue to produce sperm, which then can be recovered to fertilize by assisted reproductive technologies. So this slide just shows you how we performed that autologous grafting. These are in animals, as I said, that were chemotherapy treated. We also castrated the animals because this allowed us to distinguish the outcomes from the grafted tissue from possible outcomes that would come from endogenous testicular tissues. At each graft site, we individually sutured 4 little pieces of immature testicular tissue under a skin flap and then simply sewed the skin flap shut. And then we transplanted tissue in an experimental design that allowed us to compare graft outcomes in fresh tissue versus frozen and thawed. Of course, we're most interested in the frozen and thawed tissue because that represents our prepubertal cancer patients. The experimental design also allowed us to ask about graft outcomes when the grafts were put under the back skin versus under the scrotal skin. And finally, we introduced matrigel into some of the graft sites because we thought that that might induce angiogenesis into the grafted tissue. Well, it turned out that it didn't matter whether the tissues were fresh or frozen. It didn't matter whether they were transplanted under the back skin or under the scrotal skin. And it didn't matter whether or not we injected matrigel. In all cases, the grafts grew continuously after they were transplanted through an 8 to 12 month period of time. When we recovered the grafts, you can see that those 4 little tiny pieces of tissue have now grown substantially and merged into one large mass. You even can see a suture remaining here in one of the pieces of tissue. And when we dissected this tissue apart, we were able to release sperm, what are normal looking testicular sperm. And it wasn't just a few sperm, we were able to release between thousands and millions of sperm from every single graft that was recovered. So the next question for us is, are those sperm competent to fertilize monkey eggs and establish a pregnancy? Again, to test that question, we sent the sperm off to our friends at the Oregon National Primate Research Center who performed ICSI. And we found that indeed the sperm are fertilization competent because after ICSI, it led to the development of 2-cell embryos, 4-cell embryos, 8-cell embryos, and et cetera, all the way up to the stage of a hatching blastocyst, which is the stage when an embryo would implant into the uterus. So that's exactly what they did. They transferred the embryos into surrogate females. This happened in November of 2017. We were able to confirm pregnancies a month later biochemically. 1 month after that, by ultrasound, we could confirm that fetal development was happening normally. So at that time, we scheduled a full-term ultrasound. And the result is the birth of Grady, which stands for graft-derived baby. She was born on April 16th of 2018, and she's the first monkey baby born from autologously grafted, frozen and thawed prepubertal testicular tissues. So this is a mature technology. It's been replicated in numerous animal models, and I think that our work in the monkey establishes safety and feasibility that we believe justifies translating this technique to the human clinic. And by the way, immature testicular tissues have been frozen for thousands of patients worldwide. So I think it's our obligation to responsibly bring these new technologies to the clinic so those patients will be able to use them in the future to have biologically related children. So just a few words about options for women or girls who cryopreserve ovarian tissues. Similar to the boys, to the men, we have established standard of care ways to preserve fertility, and those are to cryopreserve oocytes that can be thawed in the future and fertilized to achieve a pregnancy by IVF. That option isn't available for all adult women because some women with a serious cancer diagnosis won't have time to undergo ovarian stimulation before they can before they start their therapy. And so for those adult women and also prepubertal girls who aren't producing mature eggs yet, we have the option to cryopreserve immature ovarian tissue, which is the primordial follicles that Sherman told us about that are located on the outer surface of the ovary. You've heard wonderful work today, both by Sherman and by Katsuhiko, about ways that the immature follicles might be matured outside the body to fertilization stage. But I'm going to tell you about the option of transplanting those tissues back into the body of a woman. This is a technology that Sherman has helped to revolutionize worldwide. It was first performed in 2004 and now has led to many live births worldwide. And I'm going to tell you about that through the lens of one particular patient. This, I believe, is the eighth ovarian tissue transplant that led to a live birth. It's the story of a woman named Stina Berkholt in Denmark, who was in early menopause at the age of 27 after she was successfully treated for Ewing sarcoma. So she had cryopreserved tissue before she underwent her treatment. When she was ready to start a family, she came back and asked to have that tissue transplanted back into her body, which was done. And the doctors advised her at that time that she should try to get pregnant as soon as possible, because in those early days, we didn't know how long the ovarian tissues would last. And so she went ahead to do that. She had her first child by IVF. And when she was ready to have her second child, she returned back to the doctor and said, I'd like to have another piece of tissue transplanted. I'm ready to have another child. But to her surprise, she was already pregnant from the originally transplanted tissue. And in this case, she was pregnant in the normal way. And that's how she had her second child. Well, now over 130 live births are published and documented worldwide. And also we know that those tissues last a lot longer than what we originally thought that they would do, including not only their reproductive potential, but also the return of hormone function. And so based on these outcomes, the American Society for Reproductive Medicine advises that ovarian tissue freezing is no longer experimental. So I'll finish up today by telling you that I have reviewed some autologous transplant technologies, both stem cell based and tissue based, that might be used to restore fertility in patients that are made infertile because of a medical treatment. But those autologous transplant technologies won't be appropriate for all patients. For example, patients with leukemia or testicular cancer or ovarian cancer could harbor malignant cells in their tissues, and it may be unsafe to put those tissues back inside their body. Similarly, transgender patients who will not experience a puberty in the gender that's required to mature their gonads wouldn't be able to undergo an autologous transplant. And finally, patients who have genetic causes of their infertility that prevent germ cells from developing, it won't do any good to transplant their germ cells because they have a genetic defect that would prevent them from developing. And so for those patients, we have to look for methods to mature gonadal tissues or cells outside of the body to produce eggs or sperm, including the technologies that you've heard about today from Amander and Katsuhiko and Sherman. So I'll finish up, first of all, by thanking our CHOSE team. This has been a wonderful collaboration, I think, with multiple different kinds of experiences. So I think we can learn a lot from each other, and we do all the time. I've tried to acknowledge the people who did the work in my lab as I presented it, but this has been a large effort over many years period of time. And so you can see that there are a lot of names who've contributed in one way or another, intellectually or directly in the experimentation. Of course, none of this work could be done without generous funding. And as Amander said, I'll also acknowledge the Luca Bella Foundation that supports our CHOSE project. And finally, I always like to acknowledge patients who both inspire the work that we do and in many cases donate tissue to our research so that we can develop next generation technologies that we hope will be available for them in the future. So with that, I'll finish up and pass the baton back to Sherman.
Well, thank you very much, Kyle. Again, that was very, very enlightening, and all kinds of questions are opened up. I've been asked by Ziv to start the question and answer, so there's an awful lot to ask. I'll make a point to Kyle and then a question. We actually have found, based on advice from Klaus Anderson, who you know, that if we take out the ovary of a leukemia patient after she's in her first remission, there won't be viable cancer cells. And we actually have transplanted 3 of those type of cases back. And one of them has 5 babies already, and one of them has 1 baby, and one of them hasn't gotten pregnant. But I'm wondering whether with prepubertal testis biopsies, which I'm assuming that should be done on every cancer patient, every male cancer patient who's prepubertal, and you have a possibility of either a tissue transplant or a SSC or a cellular transplant. How will you get rid of the cancer cells and not risk the transmission of cancer? Could it be the same thing that if they've had their first remission, that the SSCs might have survived, but the cancer cells not survived? I'm wondering if you could answer that one, what you think.
Yeah, so I'll answer your question in 2 parts. First of all, we have published in the past, both using monkey cells and human cells that were contaminated with leukemic cells, that we could sort out the cancer cells from the transplantable stem cells with 100% outcome. And so it works. But however, that is, we didn't see a cancer in the mouse experiment. So we were transplanting these tissues back into mice. I don't know that we're convinced yet that we can be 100% sure that we never would reintroduce a cancer cell. And so I would be hesitant to do that, at least in the earliest cases. We do have cases, as you described, and as have been described for transplanted ovarian tissues. We have quite a few patients who are leukemia patients who are who their cancer comes back after initial treatment, and that's when we cryopreserve the tissues. And it's possible that the malignant cells are removed from the testicular tissues in that case too, but as far as I'm aware, this hasn't been studied.
Very, very interesting. Of course, there's always a conflict with the ovary and leukemia patients. We'd like them to have had their first chemotherapy and been in remission for the reasons stated, but then that makes it impossible to do in vitro maturation. So maybe we want the ovary out just to do in vitro maturation before they've had chemotherapy. And it's a conflict. Yeah. And Amander, I direct a question to you. With the patients that have mutations that are causing the azoospermia, those male patients, is it possible, do you think, to get the germ cells and use CRISPR, or is it even legal, to get rid of that mutation so that the germ cells would function when they're replaced? Oh, you got to turn on your unmute.
Finding the unmute button. So yes, you're absolutely right. So in the lab right now with those patients' iPS cells, we are using CRISPR base editing technology. So instead of making a cut, what that does is it changes the base from the T back to a C, which will correct the amino acid. We're doing that in the lab for research purposes because we want to understand that mutation in the core domain and how it affects germ cell formation. So we need a control. And so the control is gene correction of the patient's iPS cells so that we can study germ cell development in our patient and also the gene corrected patient cells at the same time. Could gene correction be used in iPS cells for reproductive purposes? That is part of an international conversation right now. I think the first thing is that IVG from iPS cells would need to be proven safe, and governing bodies would need to agree that enough evidence has been presented that it's safe. And then on top of that, the current governing bodies that are looking at germline genome editing would also need to agree that this could be used to introduce corrected genes back into the human population. So those conversations are all going on right now. Theoretically, it would be possible, but that's where we are.
I take it right now you can do CRISPR on the iPS cells, but you can't do it on the germ cells. Is that right?
So in animal models, it's possible to do CRISPR on germ cells. So for example, it's possible to use CRISPR to alter the genome in the oocyte before fertilization. Most mouse colonies around the world use CRISPR now to introduce changes to genomes in order to study genes in development. But in humans, it's a little bit different. I think we understand very little about the early human embryo development. And so if we knew more about how DNA is repaired in early human embryo development, I think we'd have a lot more confidence using CRISPR technologies in the human embryo. So right now, CRISPR introduces mutations, off-target mutations and on-target mutations in human embryos that are not well understood. But in humans, nobody has used CRISPR technologies on, as far as I'm aware, and maybe Kyle and Katsuhiko knows more, on human eggs or even human sperm. There was one study that suggested that they had gene-edited human sperm, but they themselves were not able to repeat that result. So I think we still really don't know.
Well, Kyle's the right person to go to on this because he originally said to me, well, maybe CRISPR for iPS cells won't be so controversial, but CRISPR for germ cells in the human is very controversial. Would you comment on that, Kyle?
Sure. I'm not sure that's exactly what I said, but what I will say, and just to follow up on what Amander said, one of the limitations of doing gene editing in embryos is the number of embryos that you have. Because of course, when you do an edit, you're going to get the correct edit that you anticipate. You're going to get incorrect edits or no edits at all. And so when you have a limited number of embryos, the chances to get exactly what you want is small. And then you still have the possible issue of off-target changes. And what do you do if you have an embryo with the correction you expect? And also off-target changes where you don't know what the result of those could be. So I actually think that that's one of the advantages of using either spermatogonial stem cells. I chose to take out a slide where I would have shown this, to using either spermatogonial stem cells where you can maintain thousands or even millions of cells in a petri dish and choose out the rare event that has the change that you're looking for and hopefully minimal off targets. The challenge for doing that in humans with spermatogonial stem cells is that we don't yet know how to maintain human spermatogonial stem cells in culture. We can do it for mice, but we can't do it for humans yet. And that's where I actually think that the iPS cell route is terrific. We already know how to maintain human iPS cells in culture, as Amander showed. those cells can be edited in vitro, again, with the expected and unexpected outcomes. But if you have millions of events, you can select for the ones that are the best modified. The only thing that we don't know yet about the human germ cells is whether they're transplantable or whether they can differentiate to eggs or sperm in a petri dish. So I think those are the things that we have to work on moving forward.
I go further on that with asking you this, Kyle. We certainly need fetal somatic ovarian cells for eggs, but do we really need fetal Sertoli cells or organized for sperm?
I suspect that we do, and I think the route that Katsuhiko described, where we create those cells in vitro, again from pluripotent cells, will be the appropriate route. There's been some work on that already. I think the question is how many of all of those cells in a fetal testis, how many of those cell types do you really need to direct proper germ cell differentiation?
So you're not going to be able to inject PGCs into a recently post-pubertal male and have them really accepted or work. Is that right? You still need fetal.
Well, you need the fetal cells to get the primordial germ cell-like cells in the first place, don't you?
Yes, but then
I'll ask Amander or Katsuhiko to correct me.
Yeah, but then for going from PGCs to SSCs.
Yeah, so that approach has been described because PGC. So there is evidence that, at least in mice, that PGC LCs are transplantable. But the efficiency, well, I think there are two issues there. So basically the outcomes have been good, but those aren't ideal transplantable cells. They're not equivalent to spermatogonial stem cell-like cells. But Ishikura and colleagues recently reported that you can actually differentiate PGCLCs one step further to be what they call germline stem cell-like cells, which are equivalent of spermatogonial stem cells, I think. And those seem to have a higher transplantation potential. I don't remember that that's been reported yet in humans, but at least in mice, the two advantages of that, they can maintain those germline stem cell-like cells in culture for a long time. And also when they transplant, the regeneration potential is much higher than primordial germ cell-like cells.
But you don't see injecting PGC-like cells into a recently post-pubertal male and getting anywhere with that approach.
In mouse, it works. That's what I can say. Katsuhiko showed that if you transplant a PGCLC into a newborn mouse, which would be equivalent to a teenage boy because mice go into puberty just within a few days after birth. So if the PGCLCs that are being produced in humans are equivalent to the PGCLCs that have been produced in mice, in principle, they have the potential to produce spermatogenesis if you transplant them into a young testis.
Okay, well, I think that's a very, very important thing. That's really, really important to know.
I just want to add, though, one quick thing is what makes Kyle's work so important is that he's using a primate model, going from mouse to primate to talking about technologies that are now approaching being ready for the clinic. However, nobody has transplanted a rhesus PGCLC into a prepubescent rhesus animal that's infertile. That experiment needs to occur first.
Yes, of course. Well, you made that clear. I wonder, Amander, general question. Do you have any idea when, we talked about this a few months ago, but when this technology might be available for menopausal women or azoospermic men, available like in 5 years, 10 years, 15 years? What would you, because we've already made huge progress with the mouse with Katsuhiko's work on making fetal granulosa cells from stem cells.
Yeah, Sherman, it's really difficult to say. So maybe rather than putting a time on it, I will say that this technology will be available for use in humans once it's proven safe through preclinical testing. So, and science moves in unusual ways. Sometimes there's very rapid progress, and sometimes progress is slow. So I can't put a time on it, but once it's proven safe in, for example, the non-human primate models, then it'll start to be ready for use in humans.
And I know you've been communicating a lot with Katsuhiko about his approach. Maybe both of you can answer this, for making fetal granulosa cells from iPS cells. How difficult is this going to be to introduce clinically? It sounds like it's a complicated culture. What do both of you think about that?
In terms of the differentiation of the gonadosomatic cells, it's actually not so complicated, actually, to be honest. So, for example, the making circulatory cells, at least in the mouse, let's say. So, for example, to make the circulatory cells instead of the granular cells. So, we are currently involved in such the experiment. So, by changing the cytokines for sex determination, we do see the differentiation of some circulatory cell-like cells. So now we are trying to see whether those are functional or not. So in terms of this making somatic cells, because those are, let's say, not inheritable to the next generation, it seems to be more confident to use for the clinical purpose, I think.
So Amander, it sounds like it's not all that complicated.
Except that what Katsuhiko's work was built on beautiful reference maps of understanding mouse embryo development, sex determination, formation of the mesoderm, the intermediate mesoderm, the genital ridge. So our lab right now is building all of those maps in the human. So until we have the roadmap of how to do it and the embryo teaches us how to do it, it's not as simple as applying the mouse cocktail to the human cocktail. I'm certain there'll be differences. But the mouse has provided us the mouse has provided us a really great start.
That leads me to what was the next question is, it seems like you're using the same genes for a lot of what you're doing in the human that Katsuhiko is using in the mouse. Shouldn't it be vastly different, or is it so well conserved, this process of making oocytes?
Maybe Amanda is the right person to.
Well, the transcription factors involved in making germ cells in the human are different to the mouse. So I think that's some really wonderful lessons that we've learned just using the in vitro gametogenesis technology to understand how germ cells are first specified. So a decade ago, I would have said it's all conserved, but actually we've discovered that's not true. And we have some new work showing that there are sequences in the human genome that don't even exist in the rhesus macaque that are really important sites in the genome for binding transcription factors to promote formation of the human germline. So there are some parts that are conserved, but there are some parts that are completely different. And this seems to be a real surprise to us every time we do an experiment. So
we can't just copy Katsuhiko's brilliant work. We just have to be inspired by it. Is that right?
We're inspired by it.
Okay. Do any of you have questions for each other?
Sherman, I think that Zeev is asking us to wrap it up.
Okay, well, I think this has been fantastic, and although Amander's been very cautious about it, maybe it's 50 years from now, but I'm sure at some point this technology will be applicable to humans and help women with no eggs or men with no sperm to actually have children. And we want to thank, I certainly want to thank Katsuhiko for getting this started all on the map and for all the brilliant advice he's given me. So thank you very much, Zev, for pulling this conference together.
Thank you very much, Sherman and the Kyle and Amanda and Zeev for a very nice talk.
Thanks, everyone.
Thank you. Enjoy Sundays.
You too.
Absolutely fantastic collaborating with you guys.